Alright, so I'm going to continue on with some more vocab into the second video on genetics. Um, so again, I think we've already defined this term, but I want you to make sure you have it down as well. So a zygote is a fertilized egg, right? So fertilized egg, it's also a diploid, which really means that it has both sets, it's really of a pair of homologous chromosomes, which is not the same thing as homologous structures. Okay, homologous structures are a piece of evidence for evolution, in that we have structures that have similar shape or similar structure, but we use them differently, which implies a common ancestor. This is something similar. This really means the same gene at the same locus or same position, but could have different alleles. Alright, so let me just provide you some context. In humans, we have 46 chromosomes. 23 of your chromosomes came from your mom and 23 came from your dad. But of the 23 from mom and 23 from dad, they're actually pairs. So you have corresponding genetic information for the same traits that come from mom and from dad. So when you take those chromosomes that you got from each parent and you line them up next to each other, they have matching genetic information. It's just that mom has different alleles of those genes or different versions of the gene at the same place. The dad might have different versions. So it doesn't have to be different versions, but it could be different versions. And so if we have different versions of each gene, we have a term that we use called heterozygous, which I think you've heard of before. Heterozygous, right? And homozygous means that they're the same. So heterozygous means that we have two identical alleles for a certain gene, and heterozygous means it's two different, and homozygous means two of the same alleles. Okay, we have another term that we need to use that we couple with homozygous, right? So it's either dominant or recessive. And I'm going to just go through a little word exercise with you. So sometimes you might think of dominant as stronger, which means that maybe it overpowers the recessive one, but I don't want you to think of it that way. So dominant simply means when present, or when it's there, it shows as a trait, right? So, and again, that's overly simplified, right? What that really means is when you have a dominant allele, or you have the genetic version of DNA, then it's going to code through RNA all the way to a protein, to a trait. But of course, we know that all this transcription and translation is a long chemical process, and lots of things can go wrong there. But in speaking in generalities, when there's a dominant allele, it shows as a trait. Recessive alleles only show, so that's not supposed to become, only show in the absence of a dominant trait. Now you might say, well, of course that has to mean that you have to have two recessive letters, and if there's any dominant letter, then it shows. Well, it's not always the case, right? So this is going to be a critically important definition, as well as this, for when we talk about sex-linked genes, okay? So you need to get the right definition down now, so you don't have to revise it later, right? So for sex-linked genes, these are going to be the definitions that really help us. And so when we talk about homozygous dominant, we might think of this as big A, or homozygous recessive as little a. And if we're using A's for our genes, then heterozygous is big A, little a. And these two things are the two alleles. Remember, alleles are versions of a gene, and so we represent the gene with the letter A, and the two versions of the letter A are big and little, or uppercase, lowercase. And so we can describe this as heterozygous, in which we could also say big A, little a, which is the same thing as little a, big A, but we use a specific convention. We always use the capital letter before the lowercase letter. So we don't want to use that. We always use it this way. And you'll see why later, but that's kind of the format that we use when we name these combinations. All right, so there are a couple of things that I want to do with you on this, but I want you to recognize that these are just, all of these, right? All these combinations are simply symbols. They're symbols that represent DNA. So maybe the dominant allele, I'm just doing something hypothetical here, the dominant allele, the letter A, right, this big A could represent some kind of combination of DNA. Remember that we're talking about, like, let's do TACGGTA, right? So this is just a combination of genetic information. And of course, remember, this is just one side of the DNA, and then there's also a sugar phosphate backbone, but we don't worry about that because it's always the same sugar, deoxyribose, and a phosphate group. What's the coded information that's important for this DNA to be read into RNA or transcribed into RNA is the nitrogen bases, the thymine, adenine, cytosine, guanine, and that RNA is then going to code for the protein by putting the amino acids together in a specific sequence. And then, of course, we've got this other matching side, right? ATGCC, ATGAAT, and it's got its own sugar phosphate backbone. But what's critically important is only one side, right? So what I did in yellow here is going to be the gene that codes for that genetic information. And DNA has, or the DNA replication and transcription processes have ways of reading the direction of DNA. You might remember the 5' 3' stuff from last year. But essentially, what ends up happening is when we compare this sequence to the recessive sequence, there could be a whole variety of differences, but it could be as small as something like this: TACGC, TACTTA. And so when you look at those two sequences, what should stand out to you is that there was a change here from G to C, and that's a mutation, right? So that's a mutation. It's actually considered a point mutation. It's a substitution. Substitution. Sorry, I can't spell. All right, so substitution. And so a letter was changed in the genetic sequence when DNA was replicated, which has an impact if you remember reading codons. TACGCT will code for a different sequence of RNA, which will code for a different amino acid, if you remember the codon wheels. And so that's what is really going to make an impact. So this mutation appeared, and then we inherit that mutation, and that's what genetics is really about, is how do we inherit those different combinations. So let me give you an example to help you figure this out. These symbols help us explain data that we see. So here's a scenario. It's totally hypothetical, but it's just a scenario for us to think a little bit about. So let's say that we have two dogs, right? So we have two dogs, and they're both, let's say that they are smooth coated Chihuahuas, smooth-coated, and they have, no, let's just say that they're smooth-coated, right? So basically, let's call this short-haired, right? So short-haired, I think there's specific language if you're really into dogs and stuff, but if you're not like I am, then maybe this short hair is a better description, right? So short-haired Chihuahua that's crossed with, that's an X, crossed with another short-haired Chihuahua. What this really means is that we're going to have a fertilization event, right? So this short-haired Chihuahua is going to breed with this short-haired Chihuahua. And all of their offspring, well, their offspring have different mixes, right? So we end up with one, let's say that one of them is short-haired. We've got a second one that's short-haired. And we've got a third one that's long-haired. And so what those sequences allow us to do is make some predictions about what kinds of mixes we must have gotten out of these parents. And so in order to get a long-haired Chihuahua from this mix means that the long hair had to have been hidden behind the short hair trait. And of course, that's assuming that we know that this is a genetic thing, right? So in order to be short-haired, there has to be something that's hidden behind it in order to get an offspring that has long hair. So that means that short-haired is dominant. I'm going to do a big S and exaggerate that big S. Short-haired is big S over there as well. And then the long hair is going to be recessive or hidden. And you might want to do an L for that. But remember that these are different alleles of the same gene. So we use the same letter. So I'm going to use a little s, and I'm going to under-exaggerate my s and under-exaggerate my s. And then by combining these in different ways, we can get all of these offspring. But we need to do a little bit more background knowledge. We need to explain a little bit more background knowledge here to explain what a couple of key terms are. And so these are terms that are going to come up over and over and over again. So let's talk about phenotypes and genotypes. I'm going to do phenotype first. So phenotype is the I'm going to call it the physical characteristics, but it's not always the physical characteristics. If it's a protein that's in your cell membrane, we don't see that as a physical characteristic. But basically it's a trait, right? It is the manifestation of the genetic material. So it's what the DNA is going to do, right? And what genetic adaptation it's going to provide to you, or what you might have inherited, right? So that's the phenotype, which in this case is short hair. But there's also the term genotype, which describes the genetic makeup. So if we're talking about one gene, and for each gene you've inherited an allele from each parent, you have two alleles for one gene. And so in this case, our big S, little s is the genotype. But remember that DNA leads to protein. And that's the phenotype right here. So this is phenotype. And what is it that what has the directions for phenotypes? Genotypes, which is represented as DNA. And again, this is what we understand now. This is not what Mendel understood. So we have genotypes and phenotypes. And we need to think about this as far as understanding genetics and traits that get passed down, it's usually the phenotypes that are observations or our data. Okay? And the genotypes are the inferences that we make. It's the way that we understand how genetic material is passed down. It's our explanation of how that genetic material gets passed down from one generation to the next. So we're going to talk about one more idea and then do a few practice problems. And I know that we're at 15 minutes here, so I'll post the practice problems and we can talk about them in class. All right, so there's one other idea, and this is an idea that Mendel came up with, and now we can actually see in a lab. So he calls it, or he called it, I speak as if he's still alive, the principle of segregation. Okay, and I think you know what that word means in this case in a very scientific way. It is not, doesn't have the same negative connotation, right? So segregation is basically just separation, right? So when things are segregated, they're separated. And in biology, we call it a principle, but now an even better term for it, and sometimes it's referred to this as a law, right? This is an observation. We can see this happening. Now, right? We weren't always able to do that, but we have a better understanding of this now. So the idea here is that when organisms reproduce, right? When they sexually reproduce, so when organisms, I'm going to put this in all caps because it's a new word for us, I think. We'll review what sexual reproduction means from your notes from the previous video. When organisms sexually reproduce, the alleles segregate or separate from each other. Right? They segregate from each other, which means that they carry one allele, or let's say a single copy of each gene. It doesn't always happen, but most of the time, 99% of the time, 99+% of the time, this happens, right? And so the process by which that happens, when the alleles segregate from each other, I'm just going to give you the context of this now, that process we now call meiosis, which again you know is related to sexual reproduction. All right, so here's my example. If there's a parent that might have this genotype, it's a heterozygous individual for a particular gene, what happens is that these two alleles will actually segregate from each other. We've got a big T and a little t. So this could be in the body cell of the parent, but in the, let's say this is dad, in the sperm, one sperm might only carry that allele of this genotype. Of course, it's got thousands of other genes, right? Let's not be silly and think that this sperm cell only carries one allele of one gene of all the genes in your body, in each cell. No, it's only going to be representing here, this big T represents only this of this genotype. And there's another sperm that carries that allele. Which makes sense to you if you think a little bit about it. You only inherited half your DNA from each parent. If this was in you, if you were big T, little t, you got a big T from mom or dad, and you got the other little t from the other parent. So when you are reproducing for the next generation, you only give half of your genetic material because the other half is going to be contributed by the other parent. That's what leads to more variation in our population. And we'll go over the nuts and bolts of that a little later. So let's talk about a couple of examples here and do some Punnett Squares. So I think you remember what Punnett Squares are. So I'm going to give you a couple, and then we'll talk about some other vocab in a future video. All right, add a new layer. All right, so this is what I want you to do. I want you to cross a heterozygous male with a heterozygous female. All right, that's all you're given. And I'm going to tell you that we don't know enough yet that actually the gender of these individuals don't matter yet. And really depends on where the gene is and what gene we're talking about. But I want you to solve this or set this problem up. No, I'll take that back. I'll do the first one and then I'll pause you on the next one. So heterozygous, I'm going to use B's here. So it's always good to pick a letter that is clearly distinct when it's capitalized or lowercase. You don't have to over-exaggerate like my S's earlier. But it's heterozygous. I'm going to go say big B, little b, and it's crossed with another heterozygous. So that other parent is also going to be big B, little b. Well, we're going to do a Punnett Square, and this should be familiar to you from 7th grade. And this is I'm going to give you a term here. This is a monohybrid cross. A monohybrid cross. Means one, so this is one gene. We're going to later get to dihybrids, and you're going to see some trihybrids. So we said that these actually separate, right? So we're going to put them along the outside here, and the side, the letters on the side represent the gametes. Okay? Gametes, which, another word, are the sex cells. So if these are the sex cells from, let's say this is representing the male, the dad, these would be in the sperm cells, and this combination from the other parent would be from the mom, the female, and they would be in the eggs. And then all we do inside, what we're going to do is, I'm going to write this here, but this represents the fertilization. So what we're doing is just combining these letters. So in the offspring, we end up with a homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And so we can actually set this up in ratios to report our data, right? So we've got genotypic ratios. And so, ratios, not rations, rations. All right, genotypic ratios. So we could have, we set it up with our types here. And we can say it's a 1 to 2 to 1 ratio of homozygous dominant recessive to homozygous recessive. But to understand our phenotypic ratio, we have to set up the phenotypes. And so we need to be able to understand what's going on here. And in the simplest terms, what we know is that 3 show the dominant trait, right? This is the genetic material, or this is, show the dominant to the recessive, which is going to be a 3 to 1 ratio, because remember the definition of dominant is that there is a dominant letter present, and recessive is when there's no dominant letter present, and that's only in one scenario here. So we know that there's a 75% chance that this offspring, each offspring by these 2 parents, is going to show the dominant trait. Okay? So let me give you a couple other problems. No, I'll stop there. I'll give you problems in another manner in just a minute.